Thank you for joining CureCADASIL Association's 6th webinar in our CADASIL Connection speaker series. We have switched to a webcast format to have shorter recorded webcasts instead of a live webinar. This is an effort to be able to record more webcasts from various CADASIL researchers and across many time zones. Today's speaker is Dr. Saskia Lesnik Oberstein. Her presentation is entitled CADASIL and Notch3, Genotyping, Phenotyping, and Exon Skipping. She will cover new insights into Notch3 mutations, as well as the current status of her research group's published exon skipping gene therapy as a potential therapeutic strategy for CADASIL. I'm very pleased to introduce Dr. Saskia Lesnik Oberstein. She is a clinical neurogeneticist at the Department of Clinical Genetics of the Leiden University Medical Center in the Netherlands. After completing her medical training at Leiden University, she was selected for an MD-PhD track at the LUMC. She defended her PhD in 2003, entitled CADASIL, From Notch3 Mutation to Stroke, Dementia, and Migraine, and completed her clinical genetics training in 2007. Since completing her PhD, Saskia has remained interested in CADASIL, and she set up and chairs the Dutch Expertise Center for CADASIL Care and Research. Her research group has worked on diverse aspects of CADASIL, such as the preclinical development of Notch3 cysteine corrections as a potential rational therapeutic approach, and the discovery of a novel Notch3 genotype-phenotype correlation. For her continued efforts in the field of CADASIL, Saskia has been awarded several prizes and grants. Examples are grants from the Dutch Brain Foundation and the LUMC Mary Paris Prize for Translational Research. In addition, she was recently awarded the prestigious Innovational Research Incentive Scheme grant from the Netherlands Organization for Health Research and Development. We welcome and thank Dr. Lesnik Oberstein for her time today, as well as for her interest and commitment to CADASIL.
Okay, so hello everybody. I'm very pleased to be speaking from the Netherlands about CADASIL and Notch3 and the work we're doing here in Leiden. So I would like to start by thanking the LUMC CADASIL Research Group, who have all contributed to the work I'm going to present here, and without whom I, of course, would not be able to do this work, as well as our funding agencies, which include the Dutch Brain Foundation and the Netherlands Organization for Health Research and Innovation, but especially also Platform CADASIL, which is the Dutch and Belgian CADASIL patient organization. I'd also like to acknowledge all the departments we work with very closely within the LUMC, and of course also the European CADASIL GWAS Consortium, who has also contributed to some of the work I'm going to present here. So this is where we work. This is the Leiden University Medical Center, and we are the CADASIL referral center for the Netherlands. So most Dutch CADASIL patients visit our bi-monthly CADASIL multidisciplinary clinic. And we also have our own CADASIL research lab in the building next door, and we're connected by the walking bridge you might be able to see there. And in that research building, we also have the Notch3 molecular diagnostic lab, and that actually performs all Notch3 molecular diagnostics for the Netherlands. And our clinic and our clinical research, as well as our lab research, are very closely connected in both directions. So just a little bit about CADASIL, which I'm sure all the listeners will know, but CADASIL is a small vessel disease primarily affecting the small vessels in the brain, and it's caused by mutations in the Notch3 gene, and these mutations are highly distinctive in the sense that they always lead to an alteration of cysteine amino acid in the Notch3 protein, for which the Notch3 gene encodes. And this Notch3 protein is made in the vessel wall, and when it's mutated, in the case of CADASIL, you get aggregation, accumulation of the mutant Notch3 protein in the vessel wall, and eventually that leads to a malfunctioning of the small arteries, and CADASIL patients then develop recurrent strokes and vascular dementia. So just taking a look at the Notch3 gene and protein, so this is a cartoon where you can see in blue the exons comprising the Notch3 DNA, or in fact here the copy of the DNA, the RNA, and Notch3 RNA is 33 exons, and these encode the Notch3 protein. And what we're especially interested in is exons 2 to 24, because these encode the part of the Notch3 protein which is affected by CADASIL. And that's the extracellular part of the protein, and that consists mainly of 34 epidermal growth factor-like repeat domains, or as we often call them for short, EGFR domains. And these EGFR domains always contain somewhere between 20 and 30 amino acids, amongst which there are always 6 cysteine amino acids. And these form 3 disulfide bridges between them. And these disulfide bridges are important for the folding of the Notch3 protein. Now, this is the normal situation. What occurs in CADASIL is this situation. So patients will have a mutation anywhere in one of the exons 2 to 24, usually in exon 4. That's where most patients have their mutation. And what then happens at the protein level is that in one of the 34 EGFR domains, there will be a change in the number of cysteines within that EGFR domain. So in this case, the mutation in exon 4 leads to a change in the number of cysteines in EGFR domain 3 of the protein. So rather than containing 6 cysteine amino acids, the EGFR domain contains 7 amino acids. This disrupts the disulfide bridge formation, and this is thought to eventually lead to the Notch3 accumulation and aggregation in the vessel wall. And you can see a staining of a vessel wall here of a CADASIL patient, and this in granular brown staining is actually the accumulated Notch3 protein. So CADASIL is a progressive disease, and in the course of their lifetime, patients will develop TIAs and ischemic strokes at a varying age, and most will develop cognitive impairment leading to vascular dementia. About half of patients will have migraines with aura, which can start sort of within patients' third decade, and depression and other psychiatric symptoms occur in about a third of patients. The white matter abnormalities that are seen on the MRI scan in CADASIL patients are often very small punctate abnormalities in the pre-symptomatic years, and these progress with time, and eventually there will be widespread white matter hyperintensities, and also often lacunes, microbleeds, and also brain atrophy in symptomatic patients. One thing we cannot really predict yet is what is the disease course going to be in an individual patient diagnosed with CADASIL, or someone who has had predictive DNA testing, for example, because of a positive family history for CADASIL. So we don't know which patients are going to have a classical severe CADASIL disease course, or who is going to develop maybe a much milder disease course, where we know that some patients can still remain quite well into their 70s. This is something we would like to know more about, and we have made some new discoveries there, which I will discuss. So what I will be talking about today is that we found that CADASIL might be a lot less rare than we thought. I will be discussing the disease spectrum associated with Notch3 mutations, which varies from severe to possibly very mild. What the role is of the mutation position in determining the disease severity. And then lastly, I will just spend a few slides on our Notch3 exon skipping as a potential intervention strategy for CADASIL. So just to go back to how prevalent is CADASIL in fact? Well, we started out by looking at just the situation in our country, in the Netherlands. And when you go back to 1998, when I started CADASIL research as a young PhD student, there were just 7 Dutch CADASIL families we knew of. And what we saw actually is that once we started with diagnostic Notch3 screening in our lab, that the number of CADASIL families we were able to confirm really increased over the years. And now in 2018, we know of 230 Dutch CADASIL families, and there are very many different NOTCH3 mutations within our small country alone. So we have 55 different NOTCH3 mutations, which cause CADASIL in these 230 families. And more families are being diagnosed, at a regular basis. So this did make us think, wow, how prevalent is CADASIL if we see it in our country so frequently? And then this is a map of the Netherlands, just to show you that CADASIL families do actually live all over the Netherlands, from the north to the south, east to the west. Every red spot is a Dutch CADASIL family. And then there's one place we've surrounded with a blue circle, and that's actually Leiden, where we are situated. So when you think that the current estimates of the minimal prevalence of CADASIL are about 200 to 500,000, you do sort of wonder, is that correct? Is CADASIL in fact not a lot more prevalent than that? So what we did is we actually tried to look at this from a different angle. So rather than saying we're just going to count the number of CADASIL families we know in the Netherlands or worldwide, we decided to look at the population. And just to have a look at, if we look at a large population, a non-CADASIL population, how many typical CADASIL causing NOTCH3 mutations will we see? So what we used for that is a public database which contains the DNA sequences of almost 140,000 unrelated individuals. And this database is called the Nomad browser. And you can just fill in your gene of interest, in this case NOTCH3, and you can filter out which of all these 140,000 individuals have a typical cysteine-altering NOTCH3 mutation, which we would normally associate with CADASIL. So these individuals in this database, they are from various disease and control cohorts from all over the world, but there are no specific cohorts in there, for example, who have strokes or vascular dementia. It's lots of other types of diseases, not necessarily related to anything to do with CADASIL at all. And we don't have much information about the individuals in this database, but we do know their ethnicity. So we looked at NOTCH3, and what we expected was to find between 0 and 4 mutations in this database based on the estimated CADASIL prevalence. But to our surprise, we actually found 100 times more. So we found 450 mutations which we would normally associate with CADASIL without any doubt. So that means that on average, slightly over 3 in 1,000 individuals worldwide have a CADASIL causing NOTCH3 mutation. So how does that work? That was sort of really surprised us. So we were wondering what is going on there, because what this would mean is that currently we are only recognizing 1% of people who have these NOTCH3 mutations, namely the ones we are diagnosing with CADASIL. So what's going on with all the other 99% of people who have such a mutation? Are these people who simply have a missed CADASIL diagnosis? Or are these people who somehow remain healthy, and so you call that non-penetrance? They have the mutation, but it doesn't actually affect them. They don't get the disease. Or could these people have a much milder, later onset type of CADASIL, which is not readily recognized? So we looked a little bit further into the mutations we found in the Nomad database, and also compared this to mutations we actually find in diagnosed CADASIL patients. And then we did actually find some differences related to the distribution of the mutations across the gene or the protein, as you wish. So here are the EGFR domains, the 34 EGFR domains again of the NOTCH3 protein depicted here in pink and blue. What we actually found is that diagnosed CADASIL patients most often have their mutation in one of the EGFR domains 1 to 6. About 2/3 of patients have that. whereas a third of CADASIL patients have their mutation in any of the EGFR domains 7 to 34. And this is the case for the Netherlands, but also worldwide. When you then look at the population, so the individuals in the NOMAD database, we see something different. We see that most individuals in the population have a cysteine altering mutation in EGFR domain 7 to 34, and only a minority have their mutation in EGFR domains 1 to 6. Although I do want to point out that otherwise these mutations are completely similar, and in fact 50% of mutations we see in the population have literally been described in CADASIL patients. It's just the distribution which is different in the sense of how many mutations you find in EGFRs 1 to 6 versus 7 to 34. So this actually made us think, could it be that mutations in EGFR domain 7 to 34 might be milder or predisposed to a milder type of CADASIL, and that is why you see them so often in the population where they're not recognized to be associated with any type of particular phenotype that we know of yet. So what we did is we said, okay, if mutations in EGFR domains 1 to 6 are more severe, we should also be able to pick that up in diagnosed CADASIL patients, where those patients with mutations in EGFR domains 1 to 6 might have a more severe disease course and an earlier onset than patients with a mutation in EGFR domain 7 to 34. So that's what we went ahead and compared. We looked in approximately 200 Dutch patients and looked at the age of onset of their strokes, and we divided them into two groups, those with mutations in EGFR domain 1 to 6 and those patients with mutations in EGFR domain 7 to 34. And what we saw indeed, and here you see a Kaplan-Meier plot, that patients with a mutation in EGFR domains 1 through 6 have an earlier onset of stroke than patients with mutations in EGFR domains 7 to 34, and there's about a 12 years difference there. We also looked at the severity of the MRI abnormalities, so at the lesion load, and this is in European patients, so a much larger data set even, and we also found a difference in the lesion burden on MRI. So patients with EGFR domain 1 to 6 mutations have a higher lesion load on MRI than patients with an EGFR domain 7 to 34 mutation. So taken together, it seems that CADASIL might be much less rare than we think, because we find so many mutations worldwide, but also it seems that mutations in EGFR domain 7 to 34, which are predominant in the population, they might predispose to a later onset small vessel disease, ranging from a mild CADASIL, which we recognize, to maybe much later onset small vessel disease, which is currently not recognized to be associated with Notch3 mutations. And some patients may not have, or individuals with these mutations may not have any phenotype at all. It also seems that individuals with a mutation in EGFR domains 1 to 6, that they do almost always develop a classical CADASIL, which also explains why these are the individuals we recognize and diagnose as having CADASIL. This also means that most diagnosed or severe patients have a mutation in EGFR domains 1 to 6, and that is why any therapy we develop at the genetic level, we would want to aim to target these domains in a first instance to be able to help patients with the most severe disease. So what we've been developing in recent years is something we call an approach we call Notch3 cysteine correction, and we use a technique called exon skipping. And the aim of Notch3 cysteine correction is, in fact, to counteract the toxic Notch3 protein accumulation within the vessel wall. For example, by treating patients or individuals with a mutation in a very early phase and trying to counteract the progression of the Notch3 protein accumulation in the vessel wall in order to hopefully maintain a better vessel wall integrity and also maintain then a better blood flow and delay the onset of symptoms. So what cystine correction tries to do, and that, as the name suggests, is actually to correct the number of cysteines in a mutant EGFR domain back to the canonical number of 6 cysteines. So go from 7 back to 6, or from 5 back to 6, which I show here. So in a patient, you might have 7 cysteines in a mutant EGFR domain, and we want to go back to 6 cysteines with a restoration of disulfide bridges and normal folding of the protein. And we use an RNA editing technique for that, which we call exon skipping, and you could potentially also use DNA editing techniques like CRISPR-Cas9. So we've done some of these exon skipping experiments in patient cells. So what we actually do is we target exons 4 and 5 of the Notch3 RNA. Now, RNA is simply a copy of your DNA, and there is something called pre-mRNA, and that's the RNA you can target with this therapy. And what you actually do is you develop antisense oligonucleotides, or AONs, which are single-strand synthetic RNA molecules, which you develop to hybridize to your exons of interest. In this case, exons 4 and 5. And what these AONs do is they actually hide these exons from the step into developing the RNA into the mature RNA, which is used as a template to make protein. So here you have the mature RNA, and in this case, you can see that exons 4 and 5 are not present in this mature RNA. We have hidden them, in fact. So you just go from exon 3, you skip to exon 6. This is why this technique is called exon skipping. So it, in fact, excluded the mutation from this new RNA we've produced. And then what's predicted to happen at the protein level is that we also exclude the mutant EGFR domain, in this case EGFR domain 3, from the protein, as well as all of EGFR domain 4 and 5, part of EGFR domain 6, and part of EGFR domain 2. And what is predicted to happen is that the remainder of EGFR domain 2 and 6 form a fusion domain, which we call the EGFR2-6 fusion domain. And the interesting thing is that this domain contains exactly 6 cysteine amino acids at the exact correct position, and is predicted to allow a normal formation of the 3 disulfide bridges. So what we hope is that this protein will show normal folding. And what we have, when we try this technique in cells, in patient cells, we actually can effectuate this exon 4-5 skip in the way we've meant to. So it actually works. Our AONs do cause exon 4-5 skipping. And what we've shown through other experiments, just very briefly, is that the exon 4-5 skip Notch3 protein, which is then formed, so it's slightly shorter Notch3 protein, seems to be produced quite normally by cells and at the normal location, so at the cell membrane. And it also seems to function normally like wild-type protein does. So it has a normal signaling function for the cell. So this seemed, in that respect, promising. So what the next step we wanted to try is to see, can we perform this Notch3 exon skipping in a live organism? So what we did is we developed a Notch3 transgenic mouse, and this is a mouse which, next to its own Notch3 genes, also contains a human Notch3 gene with a CADASIL mutation. And this mouse actually does develop Notch3 aggregates in the vessel wall in the same way that patients have. And what we see is that this aggregation, that this increases with age. So here you see a staining, a Notch3 staining of a 6-week-old mouse, which is quite light. And then at 12 months, you see the brown Notch3 staining increasing. And at 20 months, the staining is even more clear. And we can also measure this Notch3 accumulation, and we can give it a score, and we can see it increase in time. So we thought, well, if we can treat our mice and we can actually cause the exon skipping to occur in brain vessels, if we inject our mice with the AONs, we wondered whether we could prevent or reduce the amount of Notch3 accumulation as measured by this Notch3 score. So what we wanted to know is if we treat our mice with our NOTCH3 AONs, do these AONs reach the brain vessels? If so, are they effective? Do they cause enough NOTCH3 skipping? And if so, does that indeed reduce or prevent NOTCH3 aggregation in the brain vessels of these mice? So we've done a number of experiments, and I'm just showing a sort of a synthesis of all these experiments. But for example, here we treated our mice with our AONs for 4 weeks. And a drawback is that to effectuate exon 4 and 5 skipping, we actually need 3 AONs at the moment, which is not ideal, especially not in a living organism. You'd prefer to go back to 2 or maybe 1 AON to effectuate your skipping. So we knew this was going to be problematic, and we did a systemic delivery, so basically into the bloodstream. And what we saw in organs, here in kidney and in brain, is that we only saw in a few mice a very little bit of exon 4 or 5 skipping occurring. So in fact, what we would call homeopathic levels of exon skipping. So that's not going to be enough by a long shot to be able to determine whether exon skipping eventually will allow for a reduction in NOTCH3 aggregation in the vessel wall. It's like giving somebody a tiny bit of antibiotics and hoping it will help. We just need to get more of the AONs into the vessel wall before we can know whether it helps. So how are we going to effectuate that? How are we going to improve our exon skipping in vivo, so in a living organism? Well, we'd already tried to see what the effect was if we delivered our AONs straight into the ventricles of the brain of our mice, so intracerebroventricular delivery. And we actually saw no significant improvement of the exon skipping, so that wasn't very helpful, unfortunately. And we think what's necessary is an optimization of the AON chemistries for delivery to brain vessels. And it's something we can look at ourselves, but actually it's mostly something we have to wait to how the field advances in terms of AON development. And then we can maybe step in again when there's better AON chemistries for delivery to brain vessels. And we do know that there are some hopeful developments out there, so we are awaiting those. And also we need to reduce the number of AONs we need for exon 4 or 5 skipping, as I mentioned before, and actually we need the help of biotech companies for that, because then we need large-scale screening of AONs, and currently we're talking to a number of companies who hopefully will be able to help us with that. So what are ongoing studies is, well, one thing we want to know is individuals in a non-CADASIL population who have NOTCH3 mutations, what is their phenotype? Do they have abnormalities on their MRI scan? Do they have a late onset small vessel disease, which is much milder than anything we would normally call CADASIL? Or are they completely healthy? So we are actually collaborating with some other research groups to look into this, and we hope to be able to answer that in the not too distant future. We are going to start a clinical study in 2019, looking a bit more closely at the NOTCH3 mutation position effect. So we will be recruiting 100 CADASIL patients with a mutation in EGFR domain 1 to 6, and we will compare these to 100 individuals with a mutation in EGFR domain 7 to 34, and we will do a very thorough characterization of these individuals and hope to be able to get a bit more of a picture of the effect of the mutation on disease severity and possibly other disease modifiers as well. And of course, as I just mentioned, we will continue working on our NOTCH3 exon skipping, and are also always open to any other potential strategies which might help CADASIL patients. So our final goals, I think, are to understand the complete NOTCH3 associated disease spectrum. We want to identify potential protective measures, so disease modifiers which we might be able to establish in the studies we're going to do. Are there other factors which actually protect individuals with NOTCH3 mutations from developing CADASIL, and if so, which factors are these, and can we help CADASIL patients if we identify these protective factors? We also would like to improve prediction of the notch-free disease severity. So if we have a better idea of to what extent does the notch-free mutation contribute to disease severity, what other factors contribute to disease severity apart from the ones we already know, such as smoking, for example, and high blood pressure, then eventually we might be able to make a predictive model which we could apply to an individual to give them a bit of a better idea what they can expect for their future disease course. And of course, we're hoping to advance notch-free genetic therapy into eventually clinical trials and something we can help patients with. But that is still very much something, and we have to see how that develops. But we are certainly doing our best for that. So lastly, I would very much like to thank CureCADASIL for giving me the opportunity to share some of the research we're doing here in the Netherlands, and for making this a transatlantic connection. And hopefully you will also be in contact with the platform CADASIL in the future, the Dutch-Belgian patient organization. And thank you to anybody who has listened to this webinar.
Great. Thank you so much, Dr. Lesnik Oberstein, for this very educational presentation. And thanks to your research group for their dedication to CADASIL research. This wraps it up. The CADASIL Connection series is made possible by volunteers of CureCADASIL and a generous grant from Global Genes. Thank you.